Welcome Junior Takis, my name is Mrs. Rama. In this video, I will be helping to prepare the grade 12s for the final exam. In this video, I'm going to be looking at the Feb-March 2017 paper 1, question 3. I have removed parts of the question 3 that involve human impact, as human impact is not in the matric exam anymore. So this video might be quite short. Right, before you watch this video, grade 12s, make sure you print the question paper and have the memo with you at hand. Right, let's get down to the question. Right, now, question 3.1. An investigation was carried out to determine the effect of auxins on the growth of coleoptiles. Now, coleoptile is a young shoot. The procedure was as follows. The tip of one coleoptile, a young shoot, was removed and placed on a block of agar jelly as shown in the diagram A. So that you can see here, there's the coleoptile. There's a coleoptile, there's a tip that's removed, and there's a tip placed on agar jelly. That's diagram A. After 2 hours, the agar jelly was placed on the cut surface of the original coleoptile as shown in B, as you can see here. Then, the coleoptile was covered with a black box, as you can see here in C, and allowed to grow for 2 days, as shown in diagram C. 3.1.1. They ask you, explain why the tip of the coleoptile was placed on the agar jelly for 2 hours at the start of the investigation. So can you see this picture here? They're asking you why this happened. Now, let's look at it. At the tip of the coleoptile, from your knowledge, you know that there's auxin present in that tip. Now, for 2 hours, we are leaving that tip on that agar jelly because we want that auxin from the tip to diffuse into that agar jelly. Now, hormones move very, very slowly. And so that's why we are left the tip on the agar jelly for 2 hours to allow the hormone to diffuse slowly into this agar jelly. Right, so 3.1.2. Describe what occurred in diagram C here to cause a coleoptile to bend even though no light is present. So I know what you want to do. You want to immediately talk about phototropism, but look, I like this question. It's a very lovely question. Can you see? This coleoptile is in a black box, so there's no light. So what actually happened is this auxin diffused from the tip, went into the agar. Can you see they put the agar jelly halfway? So on this side here, on this side, there was more auxin. And there's more auxin on the left side. And that will cause more cell elongation, more growing and bending towards the right. Here you don't talk about light at all. Here you're talking about right or left. I really like this question. Right, so this is a very lovely question. 3.1.3. Describe a control for this investigation. Now, if you look at your aim here on the top, your aim is to determine the effect of auxin on the growth of coleoptile. So what are you measuring? The effect of auxin. So to set up a control, you have the experiment exactly in the same way, but you take away the auxin. Right, let's look at the memo. Yes, 3.1.1. Why was the cut tip put on the agar jelly? To allow time for the auxin from the tip to diffuse into the agar jelly. Remember, hormones move slowly. 3.1.2, we spoke about the growth. Auxin from the agar jelly diffused into the coleoptile on the left side only. We said that. This will stimulate growth and there will be uneven growth towards the right. Remember, there is no light we're mentioning. And what will the control be? Remove the agar completely or without any auxin. Okay? And place it in the black box. So set it up exactly as you have to, but there's no auxin now in this experiment. Right. That was question 3. I really like that question. It's a very nice question. Right, let's look at 3.2. The diagram below represents a typical Snellen chart that is used to estimate visual acuity, which is ability to see clearly. The extract explains how a Snellen chart is used. So I'm sure you must have seen this when you go for your eye test. A person whose visual acuity is being tested stands 6 meters away from the chart. One eye is covered when he or she reads the letters of each row out loud, beginning at the top. The smallest row that can be read accurately indicates the visual acuity in that specific eye. Visual acuity is expressed as a ratio, and the line designated 606 is the smallest line that a person with normal acuity can read at a distance of 6 meters. When a person is having his or her visual acuity tested to obtain a driver's license, special equipment is used that can present the letters in different patterns, arrangement, and sizes. Okay, now 3, 2, 1. A person is only able to read accurately to the end of line 5 on the Slannon chart. So, yeah. What is his or her visual acuity ratio? So there's it there. 6/12. Easy. 3, 2, 2. Explain why and reason why the special equipment can present different patterns, arrangement, and sizes of lenses is used when testing eyesight for a driver's license. This means that this pattern always changes every time you go for your learner's license or for eye test. And the reason for this is we don't want people to be able to memorize the order. And then when they go for their license, they can actually memorize it and cheat in their learners or their eye tests. Okay, and then that way you're not actually testing visual acuity. Right, let's see what the memo says. Yes, we said 6 or 12, and then people cannot learn the pattern arrangements and they cannot cheat. So that was also a nice question. This is a very short question because, as I said, I removed all the human impact questions as that's not relevant in grade 12 anymore. Right, thank you for joining me and watching this video. Join me and watch all my other videos as well.